story of the method. The genome of genetic material of any organism is made out of DNA. Each organism has a unique DNA square which is composed of base. If you know the square of the base in any organism, you have identified its unique DNA fingerprint or pattern. Determine the order of base is Kelly square. Whole genome square is a laboratory product that determines the order of base in the genome of any organism in one process. With the invention of the chain termination method, the squares became a reality. Frank Sager and his colleagues at the Medical Research Council Laboratory of Molecular Biology in Cambridge, United Kingdom, proved this. There were no way for reading the genetic code before this. Even in the small of genome, therefore, Fred was true beyond. Scientists conduct whole genome square for main step this: DNA sharing, DNA barcoding, whole genome square, and last step data analysis.
Next, in 2030, CDC began using whole genome sequencing to detect outbreak caused by the deadly bacterial Listeria. Since then, this method has allowed scientists to detect more clusters of Listeria illness, solve more Listeria outbreak while there are still small, and link ill patients to likely food sources and identify new food sources for Listeria such as caramel apple and ice cream.
Okay, now I'm going to talk about application of whole genome sequencing Pacbio. There are 4 applications. First, we have variant detection. Variant detection is a highly precise single molecule long reads enable you to confidently catalog variants of all types, from SNVs to structural rearrangement. Second, de novo sequencing. De novo sequencing is a most complex genome can be efficiently and affordably assembled using hi-fi reads up to 25 kilobytes. Third, we have genome structure. Genome structure is a hi-fi data provide information about genome structure that short-read technologies frequently omit. Even the genome most repetitive region generate long reads. And lastly, we have epigenetics. Epigenetics is a hi-fi sequencing enable you to record epigenetic changes during a routine sequencing run without the need for special chemistry or preparation.
Advantages of whole genome sequencing Pacbio. First is consider the impact of the longer rate. The SMRT method of sequencing provides a number of advantages. First, consider the influence of longer rates, particularly for the novel genome assembly, while traditional next generation sequencing can give extensive genome coverage. Short read lengths and amplification biases might result in fragmented assemblies when a complex, repetitive, or poorly amplified region is encountered. As a result, poorly amplified GC-rich and GC-poor regions are particularly prone to poor quality sequencing. Resolving fragmented assemblies necessitates more time, the bench, and more sequencing. The read set will span many more repeats and missing bases as a result of including the longer rates from SMRT sequencing runs so automatically, bridging many of the gaps and reducing the completion time. The second advantage is consider the DNA methyl transferases. For example, this can exist as standalone objects or as components of restriction modification systems. Because of the shift in DNA polymerase kinetics as it passes along the template molecule caused by epigenetic alterations, they methylate very small sequences, motifs that can easily be detected from SMRT sequencing data. Because the kinetics are changed, the timing of the fluorescence colors are noticed change, allowing direct detection of epigenetic modifications that are normally only in fact and avoiding the need of enrichment or chemical conversion. The last advantage is consider the precision. Remember that SMRT sequencing is much less accurate than other next generation sequencing platform. SMRT sequencing is less accurate than other next generation sequencing technology, which has recently been disproven in multiple ways. To begin with, a direct comparison of multiple ways to detecting genetic polymorphism revealed that SMRT sequencing performs similarly to other sequencing technologies. Second, utilizing SMRT sequencing in tandem with other technologies to assemble a full genome has proven to be as trustworthy and accurate as more traditional procedure.
Okay, next, for disadvantages of whole genome sequences Pacbio. We have 5 disadvantages for whole genome sequences Pacbio. First, expensive sequences. The price of sequences is almost 365,000 US dollar, and the second is a large amount of starting material required for a library preparation, and third, high error rate at a single pass, and fourth, shorter rate in the CC method 25 kb, and lastly, generate duplicate reagents in mtDNA.
The case study and article published involve the whole genome sequencing Pacbio. For the first one, the article that have been found is whole genome sequence data and analysis of Saccharobacillus sp. ATSA to isolate from kimchi cabbage seed, and it was found at Gyeongbok province in the Republic of Korea. For the second one is draft genome sequence data of Bacillus subtilis strain 9407 isolate from healthy apples in China. And for the last one is Feng Xin produced by Bacillus subtilis play a major role in the biocontrol of apple ring rot disease in China.
Psychological, biochemical, and other phenotypic features were initially used as species identification markers. Serotyping was one of the first methods for discussing bacteria based on antigen and antibody reaction on a species and subspecies in the 1930s. Page typing systems such as those for Staphylococcus spp were developed later in the 1950s to be even more discriminative. This approach have been used to track down the source of infection since the beginning. Gene regulations, control and processes of cellular differentiation and morphogenesis resulting in the formation of several cell types in multicellular organisms, each with its gene expression profile. Although Maxim, Gilbert and Sanger created the first DNA sequences method, which allowed researchers to determine the exact base pair sequence of a DNA fragment in 1977. They did not widely use it in microbiological typing then.
Today I will discuss about whole genome sequencing. WGS stands for whole genome sequencing and is a method for examining complete genomes, identifying inherent illnesses, defining the mutation that cause cancer progression, and tracking disease outbreak have all benefit from genomic information. Whole genome sequencing has become a valuable tool for genomic research due to rapidly failing sequencing press and the ability for today's sequencer to output massive volumes of data. Despite research improvement in medical knowledge, doctors are still baffled by some disorder, and they are turning to whole genome sequencing to solve the puzzle. The method of determining the DNA sequence of an entire genome is known as whole genome sequencing. When there is a genetic defect at the core of an illness, whole genome sequencing is a brute force method to issue solving. Several laboratories now offer genome sequencing and analysis and interpretation services. Whole genome sequencing process. For 1, DNA extraction. Researchers tear open bacterial cells on an agar plate using chemical that spread them open and release the DNA. After that, the DNA is purified. 2. DNA sharing. DNA is cut into short, non-large segment added with enzyme, calent molecules cancer, or by mechanical description. For 3. DNA library preparation. Using a technique known as polymerase chain reaction, scientists make numerous copies of each DNA fragment. PCR. A DNA library is a collection of fragments created by a PCR machine. For 4, DNA library sequencing. A sequencer is used to load the DNA library. Its result is referred to as a DNA read, which is the combination of nucleotide A, T, C, and G that make up its unique fragment of DNA. And lastly, DNA sequences analysis. The sequencer generates millions of DNA readings which are assembled in the correct order using specialized computer programmers, much like a jigsaw puzzle. The genome sequence will contain millions of nucleotides in one of a few large parts. Is ready for further study once it is complete. That's all for me. Thank you.